the field's in a very interesting state right now. So for many years, we have been approaching this problem by using physically based models, in particular the Rosetta program developed in my group and many collaborative groups around the world. And the basic principle here is that proteins fold to their lowest energy states. So if you want to design a new protein structure, the challenge is to design an amino acid sequence whose lowest energy state is that structure. And in essentially everything I'm going to tell you about today, we're not really using any information from naturally occurring proteins. We're not starting from naturally occurring proteins. We're starting from scratch. So we make up a brand new structure on the computer, and we search for a sequence whose lowest energy state is that structure. And but deep learning, as you just heard, is really coming online. And so towards the end of my talk, I'll show you how deep learning is now starting to transform how we do protein design. So one of the problems that we have been working on for a while and recently made considerable progress on is the general problem of, given a protein structure of interest, design a small protein which binds very tightly to it in at a site of interest. And I'll illustrate that here with the coronavirus receptor binding domain, which is shown in cyan here. So at the beginning of the pandemic, we used these methods to design proteins, small proteins. So these are completely made up proteins. We generate a very large library of scaffolds, dot them against the structure, and then redesign the sequence at the interface for high affinity binding. And examples of what comes out from this are shown here. So on the right, you see here's a small protein. It's 55 amino acids. Turns out when we made it in the lab, it doesn't melt at 95 degrees. And you can see it makes these really shape and chemically complementary interactions with the virus. Let's see. Okay, so after we made the protein, we were of course curious to see whether the structure, the way it bound, was the way we had designed it to bind. And so this is now a cryo-EM structure. You're looking down at the top and you see 3 copies of the small protein binding. And if you superimpose the computational design model and the cryo-EM structure, you can see they're virtually identical here, even at the level of the side chains. So we can design proteins that bind to almost arbitrarily selected protein targets at particular sites on their surfaces. So as escape variants started coming out, we sort of took the design to a higher level, and we designed this trimeric structure here, which holds 3 of these receptor binding domains in position where they can engage 3 different receptor binding domains on the coronavirus. And this protein is a very potent inhibitor of all the both wild-type coronavirus and all the strains that have been tested so far, with 100 picomolar neutralization of Omicron and the other variants. It's also quite effective in animals when administered intranasally. And we're hoping this is now in sort of preclinical studies, and we're hoping that will be in clinical trials by the end of the year. So these methods for designing binders to arbitrary protein targets can obviously be applied not just to pathogen proteins, but to naturally occurring receptor subunits too. And so we've been sort of going through systematically and designing binders to growth factor receptor subunits and cytokine receptor subunits. And we can now use these binders and scaffold them in higher order structures like that trimeric structure I showed you, to bring together receptors in new ways, cell surface receptors. And this shows an example. This is a collaboration with Yossi Schlesinger's group. Basically, we took a small protein design to bind the FGF receptor and design these hexameric scaffolds. And we fused the FGF receptor on the hexameric scaffolds. We get really pronounced elicitation of FGF signaling. The cool thing is we can change the valency and the geometry with which these things are displayed, and we can combine different receptor binding domains to bring different combinations of receptors together. So we're really excited about uncovering a lot of new signaling biology with perhaps therapeutic potential here. So those were pretty static structures. We can also design proteins which undergo conformational change. And so here we've designed a molecular device which has an off state and an on state. And in the off state, it does not emit light. In the on state, it does emit light. And we have in the off state a binding domain that can't bind to a target because it's caged. In this case here, we use the binding domain that I just told you about for the coronavirus. And in the presence of the target, the coronavirus, the thermodynamics of binding drive the opening of the cage, which leads to reconstitution of luciferase activity and luminescence. This works quite well. You can see here that so the sensor is dark until coronavirus protein is present, and then there's this a rapid increase in the emission of light. Another way in which we can use sort of switchable systems is for distinguishing cells based on combinations of surface markers. So for cancer therapy, you frequently would like to be able to target cells that have some combination of surface markers and not just based on single markers, because there might not be a single marker which distinguishes tumor cells from normal cells. And so here what we can do is target one component of a switching system to one marker, another component to another marker, and only if they're both present will the system switch. And you see this on the right column here. I have I'm showing a more complicated version of that, where here we have one A and B, not C logic. So we have the two components of the switch targeted to cancer markers, and then we have the third component that is that's targeted to a healthy cell marker. And that third component actually shuts down the switch. It's basically biochemically, it sequesters one of the components. So in this way, we only get activation of the switch on cells that contain two of the markers and not all three. And that leads to CAR T cell activation and cell killing. We've also been designing proteins which assemble into larger structures. And so here on the left, I'm showing what you're seeing are design proteins in green that bind to the constant region of antibodies, which are shown in yellow, and which cause the antibodies to be wrapped up into nanoparticles. So we have icosahedral nanoparticles here, we have octahedral nanoparticles here, tetrahedral ones here, and these are depicted here. And what's very interesting is when we take antibodies that bind to cell surface receptors and incorporate them into these nanoparticles, we get a pronounced enhancement of agonistic activities. So for example, for a death receptor antibody, the plot on the right illustrates sort of an enhancement of cell killing. So we can also develop these for delivering biologics into cells. So here we have the tetrameric protein and the antibody, and you can see that there's this empty face here. This is like the corner of a cube, since this is an octahedral structure. So Aaron Yang basically designed a plug which has three arms like this, and it docks perfectly in here. And here you see the cryo-EM structure of this. And I'll have another view here. Here's the cryo-EM structure. You're looking down the threefold axis. That's where the plug is. And you can see that hole is now filled. This is down the fourfold axis. This is that tetramer that brings the antibodies together. And what's really cool is that Aaron's designed these to be pH dependent, and they come apart at very modest, at just barely below neutral pH. So these are different variants, and these ones are coming apart by at about above pH 6. So we're pretty sure that in the endosome, this plug would come off, and then the contents would come out. And so Aaron's developing these now for delivery of RNA and other applications. So the applications of protein design aren't limited to the medical realm. Here we have Nate Ennis in the group has been designing proteins which bind to chlorophyll dimers in exactly the same orientation they are in photosynthesis, in the photosystem. And so, they're excitonically coupled. And so what he and Shunji Wang then went on to do is to build these octahedral structures where there are two chlorophyll molecules on each edge of the octahedron. And so we're very excited about these as starting points for new light harvesting systems. So a grand challenge for protein design for some time has been enzyme design, and Andy and Chris Norden took on the challenge of designing new luciferases from scratch. So the way they did this was to design proteins which had pockets which were very shape complementary to this oxyanion intermediate. And I'll show you how they did that. So this is the first introduction of deep learning into my talk. So Chris had developed methods for designing large numbers of scaffolds with pockets roughly this right size for this substrate. And then Andy used matching methods we've developed to basically place the ligand into the pockets such that it could make the anionic group could be stabilized by an arginine residue. And the net result are designs that look like this, where you have a designed protein that is very shape complementary to the ligand, and which has an arginine here stabilizing the development of the negative charge. So this was very successful, and you got this. I should say this is a substrate that naturally occurring luciferases haven't been optimized for, and here you see that this sort of quite strong luminescence. It's readoutable with an iPhone. I can't see it very well here, but if you compare the kcat over km to Andy's designs to the naturally occurring luciferase, they're pretty comparable, which is pretty neat. But Andy's design, so the naturally occurring luciferases have these big wide open pockets, and so they work on many different substrates, which makes it hard to multiplex them because they're kind of promiscuous. Andy's design, in contrast, since it has this very shape complementary pocket, is very specific for the intended substrate. And so this is neat. So now Andy's designing hopefully similarly specific enzymes for these other substrates, which should really enable sort of multicolor luminescence sensing in the sort of similar way that people use multicolor fluorescent protein. So a longstanding challenge in protein design that Li, Shenji Wang, and Una Natterman took on is the design of proteins which assemble into crystal lattices. So this is here what the challenge is to design the crystal contacts. So the way they did this was to use a hierarchical approach, starting with monomers, design them into oligomers, and then into polyhedra, and then design those polyhedra into crystals by designing interfaces between them. And the results are quite beautiful. Here are 3 different crystals that they designed between these polyhedra with these quite well recapitulated crystal interfaces. And you can see the theoretical and the observed Sachs profiles are also very, very similar to each other. So the final example I'll give you of traditional protein design is the design of molecular machines. And this is work by Alexis Courbet. And so he designed axles and rotors that can rotate around the axles. And he also developed methods for assembling the rotor rings around axles, as shown here. So he designed a series of different axles and rotors, and some of these are shown here. And then he assembled the axles on the rotors. And when he does this, you can then sort of compute the energy landscape with Rosetta by rotating around the ring around the axle. And in some cases, you get pretty flat energy landscapes. And in those cases, when you look in the cryo-EM, the rotor is looks like it's sort of rotationally diffusing around the axle. In other cases, there's deeper minima, like this case, and I'll show you that one in a little bit more detail. Here, Alexis was able to distinguish 2 different rotational states. In this system, he's actually assembled 2 rotors on one axle. It's on the way to like the protein car, as you can tell. So here you can see in one state that the 2 wheels are kind of staggered relative to each other, and here they're more eclipsed. So now the challenge is to couple this to an energy source to drive rotation. Okay, so now I'm going to talk about deep learning. And so we've been developing, and obviously, others have been developing methods for predicting protein structure from sequence. And so the first way we set about to use these models to design proteins was to basically feed in to the network completely random sequences, and then which aren't predicted to fold to any structure in particular, and then to optimize those sequences until the network thought they folded to some structure. And this is what this looks like. So starting with the random sequence, this is a contact map, so residue by residue, and you can see there aren't really many predicted contacts for these random sequences. So take the random sequence, put in the network, says it doesn't look like it folds up to anything. Now we're going to optimize the sequence by Monte Carlo and accept sequence changes that are predicted to make the protein fold into some structure. We don't specify what that structure is. By the end of that, we end up with these very strongly predicted sets of contacts, which correspond to folded structures. And we've made a bunch of these now in the lab, and it's really cool. These, we call them hallucinations because it's kind of like this kind of the network's idea of what ideal protein should look like, because it's just kind of making it up out of noise. And you can see the hallucinated structures are very close to the crystal structures. So to show you how far things have come, this was a rotation project starting in January of this year, so I guess 3 months ago. So in this case, 2 rotation students, Sam and Elias, were charged with the problem of doing this hallucination, but basically rather than just randomly generating a sequence, after randomly generating it, tandemly repeating it, in this case 15 times, and then predicting the structure and optimizing just that one repeat unit and propagating the changes all the way along. And what they came up with is this 1050 residue protein, which has 15 repeats of a strand helix motif. And when they made the protein, they found that it expressed well and assembled. And then Alexis Courbet solved the cryo-EM structure of this, and it basically came out exactly right. And so, I mean, this kind of so rotation in 10 weeks, you can kind of do a lot in protein design these days. And so these will be the rings for the next generation of protein machines, because they have kind of ideal properties for that. So far as I've described it, there isn't really an immediate way to put function in, but what we can do is modify this hallucination procedure to add to the loss not only how well the sequence folds, but the presence of a desired functional motif. And so I'll just illustrate that here in the context of vaccines. So this is the RSVF protein, and it has motifs which are recognized by neutralizing antibodies. So a goal of vaccinology is to make small proteins which present these structures in small proteins, so you can really focus the immune response in those places. So here is here's one of those cases. This is this motif here, this site 5 motif, and here are some design proteins that Zhu Wang made using this hallucination approach, which are where he's favoring sequences that will recapitulate this motif. And you can see that these structures actually do contain the motif so the hallucination process worked to basically, in that case, basically what we're doing is we're specifying it's kind of a functional motif and then just asking it to fill in the rest. It's kind of like specifying the ears of a cat and then asking the network to fill in what the rest of the cat should look like. And what's really cool is we just made these proteins in the lab, and they bind the antibody. They're also well-folded proteins. So we have a second approach for function design, which is analogous to in-painting approaches used for images. So you're probably all familiar that if you type into a word processor, at a certain point, it will try and complete your sentence. And so we took the Rosetta fold model that we've been optimizing and developing for structure prediction and trained it to not only predict structure from sequence, but also to predict sequence from structure, and also to be able to, we gave it training examples where parts of the protein were missing and had to fill it in. So now Rosetta fold is like a general, so you're familiar with language models for natural language, and language models for amino acid sequence have become popular. But now we have this general model for protein structure and sequence. So we can specify, say, the sequence and structure for part of the protein, and then the network will just fill it in, like auto-completing your sentence. And so here's an example of this. Joe Watson and David Jurgens, they basically took a little motif that binds iron and asked the network just to fill it in to complete it out to make a full protein. And they've just made a bunch of these proteins. They're very stable, and they indeed bind metal. So I think this would be a very powerful method for protein function design. And then I just wanted to close on, as I said, we've been developing RosettaFold. We've been improving it. It's now pretty much as accurate as AlphaFold, and we've been using a combination of RosettaFold and AlphaFold to predict structures of protein-protein assemblies. And so so we basically took the yeast genome, made all pairs of proteins, and then predicted whether they interacted, and if so, how they interacted. And these are just examples of structures that we were able to solve using this approach that fall into different biological processes. And we could not only identify pairs that interact, but also sets of proteins that interact. And we were able to assemble these into higher order complexes. And this was really neat. So there were a number of cases like this, where this is the assembly that puts GPI anchors on proteins in eukaryotes. And we found Tamara Hendrickson, who's the world's expert on this complex, and she sent her the model, and she pointed out all these interesting things, like there was this catalytic dyad that was adjacent to a putative peptide substrate recognition channel that we hadn't really noticed. And so these kind of models can provide all sorts of insights into biological function. So I've had lots of really, really fantastic collaborators in all this work. So Longjin and Brian developed these general methods for designing binders to arbitrary targets. Andrew Hunt and Brett Case and a whole host of people were involved in sort of making these multimeric versions. Keija Wu designed the one I described to you, and then testing them in a variety of animal and cell culture models. Natasha and Thomas are doing the superagonist development. The sensing and logic work was done by Alfredo and Andy. That was the sensors, and then Jillian and Ruffy and Mark LaJoy did the work on the cancer cell targeting. The 3-component closed nanocage work is done by Robbie, Aaron, and Andrew Borst. The design of luciferase done by Andy and Chris Norton. The design of protein crystals by Zhe Li, Shunji Wang, and Una Natterman. The design of molecular machines, those rings that move on rotors, is done by Alexis Corbet and Jesse Hansen. The protein hallucination of just the original proteins, the monomeric proteins, done by Yvonne, Sam, and Temuka, and Guy Montaglioni's group helped a lot with solving structures. Alexi and Elias and Sam designed those giant rings, and the methods were developed by Basil and Lucas. The functional site scaffolding, the constrained hallucination work was done by Zhu Wang, Sydney, and Doug, and it's been a great collaboration with Sergei Ovchinnikov. And David Jurgens and Joe Watson did the in-painting sort of autocomplete approach. RosettaFold is really the work of Minkyung Bak working with Frank DeMaio. The protein interaction prediction work is done by Chen Kong, Ian Humphreys, Aditya, and Minkyung. And